السلام عليكم ورحمة الله وبركاته. أنا يسرى صادق، مدرس باثولوجيا إكلينيكية. محاضرتنا النهاردة هتكون عن Immunoglobulin Proteins Structure and Function. Immunoglobulin proteins. They are the critical ingredients at every stage of humoral immune response. When expressed on the surface of resting B lymphocytes, they serve as receptors, react with specific antigens, and initiate a series of events that lead to B cell activation, clonal proliferation, and generation of plasma cells. But when secreted, they serve as antibodies, traveling through the tissue fluids to bind to specific molecules that triggered their production. Structure of immunoglobulin. Immunoglobulins are glycoproteins that are composed of 82% up to 96% of polypeptide and 4 up to 19% carbohydrates by weight. Every immunoglobulin is made up of two different types of polypeptides, the larger one called the heavy chain and the smaller is called the light chain. Each immunoglobulin contains equal numbers of heavy and light chains. The chains are held together by non-covalent forces and also by covalent interchain disulfide bridges. The heavy and light chain are both composed of number of domains, each of which is 100 of 110 amino acids and contains a single interchain disulfide bond. The sequences of these domains vary widely in both heavy and light chains in the N-terminal domain, whereas the sequences of the other domains remain constant. So, the N-terminal domain in heavy and light chain polypeptide is called the variable region. Other domains are termed the constant region, like variable heavy or variable light here, constant heavy or constant light. Light chain contains only a single constant light domain, but heavy chain contain three or more domains, which are numbered as constant heavy 1, constant heavy 2, beginning with the domain closest to the variable heavy chain. The pair of variable light and variable heavy domains form a single antigen binding site. This is the structure of the immunoglobulin. The inner part called the heavy chain and the outer part called the light chain. The yellow part here called the variable domain and the blue one constant domain. The region between the variable light and the variable heavy domains called the antigen binding site. The upper site called the N-terminal end and the lower part called the carboxy terminal end. The protein has a Y-shaped configuration. The region at the base of each arm in the Y located between the constant heavy 1 and constant heavy 2 domains is called the hinge region. This offers the flexibility to the immunoglobulin, enabling the two arms to move freely with respect to each other. Enzymatic digestion products of the immunoglobulin. There are two enzymes can digest the immunoglobulin protein. First one, called the enzyme papain, can cleave the hinge region on the N-terminal side of the inter-heavy chain disulfides, resulting in three fragments of roughly similar size. These fragments are called two Fab fragments, antigen binding fragments. Each fragment has monovalent antigen binding activity. And FC fragment, the fragment which contains the carboxy terminal portions of both heavy chains held together with disulfide bonds. Most of the secondary biological activities of immunoglobulins are determined by the sequences in the FC region of the protein. Also, it is the site that is recognized by FC receptor on many cells. Here, when the enzyme papain can cleave the immunoglobulin region, it results in FC portion and two Fab fragments. The second enzyme called pepsin, which cleaves on the carboxy terminal side of the interchain disulfides into a single large fragment called Fab2 fragment. It has divalent antigen binding activity, and the other fragment called the FC region is extensively degraded by the pepsin and usually doesn't survive as an intact fragment. This is the enzymatic digestion of immunoglobulin protein by the pepsin, resulting into large Fab2 fragment and Fc fragment. Light chains types and subtypes. All light chains have protein of molecular weight about 23,000 but can be classified in two types called kappa and lambda. The constant region of all kappa light chains produced by an individual are essentially identical. In contrast, one individual may express as many as 6 slightly different forms of lambda constant region. All are functionally identical. The ratio between kappa and lambda chains production in a human is usually 2:1. Heavy chain classes and subclasses. The heavy chain polypeptide ranges in molecular weight from 50 to 70,000. Human express 5 different classes or isotypes of immunoglobulin heavy chains, which differ in their constant heavy region sequence and in their physical and biological properties. The mu and the epsilon chains are made up of 5 globular domains, 1 variable and 4 constant heavy, while the gamma and alpha chains are made up of only 4 domains. All of the heavy chains in any immunoglobulins are identical. The 5 classes are immunoglobulin M contain mu heavy chain, immunoglobulin E contain delta heavy chain, immunoglobulin G contain gamma heavy chain, immunoglobulin A contain alpha heavy chain, and immunoglobulin E contain epsilon heavy chain. The gamma and alpha classes are further divided into subclasses like gamma 1, gamma 2, gamma 3, gamma 4, alpha 1, and alpha 2. The corresponding immunoglobulin classes are immunoglobulin G1 and G2. Normal individuals express all immunoglobulin classes and subclasses because each is encoded by separated genetic locus. Membrane and secreted immunoglobulin. Immunoglobulin can exist in either membrane-bound or secreted forms. The membrane form always their heavy chain contain an additional carboxy terminal sequence of about 40 amino acids. This sequence consists of highly acidic region of 12 up to 14 amino acids, followed by a hydrophobic sequence of about 26 amino acids. The hydrophobic portion is the transmembrane component that anchor the heavy chain into the cell membrane. The secreted immunoglobulin lack the terminal transmembrane segment. In immature B lymphocyte, membrane-associated immunoglobulin molecule recognize antigens, but this recognition is not enough to activate the B cells. The immunoglobulin molecules are non-covalently attached to two other proteins called immunoglobulin alpha and immunoglobulin beta that make up the B cell receptor complex. When the immunoglobulin receptor recognizes the antigen, the associated protein transmits the signal to the interior of the B cell that initiates the process of B cell activation. Framework and hypervariable regions. Variable region, which found within the N-terminal domains of light and heavy chains, mediate antigen binding and are the most heterogeneous portion of these proteins. Variable regions with any single group are not uniformly variable across their entire 110 amino acids. These consist of relatively invariant stretches called framework regions of 15 up to 30 amino acids, separated by shorter regions of extreme variable called hypervariable regions that are each 9 up to 12 amino acids in long. Variable heavy and variable light regions each contain 3 hypervariable regions. Antigen binding is mediated by interaction that involve the amino acid in the hypervariable regions of each chain. So, the sequence of these regions are that primarily determine the antigen specificity. Hypervariable regions are also called complementary determining regions, CDRs, that are designed CDR1, CDR2, and CDR3. CDR3 is usually the longest and the most variable of the three. G-chain and secretory component. First, we will talk about the G-chain. The secreted forms of immunoglobulin M and immunoglobulin A exist as polymers of the basic 4-chain unit that include a single additional polypeptide called G-chain. It is a small molecular weight, about 15,000 acidic protein that is structurally unrelated to heavy or light chains, but is sensitized by the plasma cell. Its function to facilitate the immunoglobulin polymerization. Secretory component. It is a single polypeptide with molecular weight about 70,000. It is associated only with immunoglobulin A and it is found in body secretions. Its carbohydrate content is high. Its amino acid sequence is invariant and show no resemblance to a G chain or to any immunoglobulin polypeptide. It can exist either in free form or bound to immunoglobulin A molecule. Secretory component is not formed by lymphocyte but by the mucosal epithelial cell that overlie Perez patches and other submucosal lymphoid tissue. The function of secretory component is to facilitate transepithelial passage of immunoglobulin A. In extracellular fluid, we found immunoglobulin A dimer associated with both G chain and secretory component. Secretory component facilitates the transepithelial passage of the immunoglobulin A, and when it reaches the lumen, immunoglobulin A is separated from its secretory component. Carbohydrate function in immunoglobulins. Significant amount of carbohydrate are present in all immunoglobulins in the form of simple or complex sided chains bound to amino acids of the polypeptide chains. Their function have an important role in secretion of immunoglobulins by the plasma cells, have a biologic function associated with the constant heavy region. In general, carbohydrate is found in secretory component, G chain, and constant region of heavy chains. It is not found in light chain or variable heavy regions. The higher content is found in immunoglobulin M, immunoglobulin D, and E, followed by immunoglobulin A and G. Fc receptor. Many cell types that cannot sense immunoglobulins are able to absorb circulating antibodies into their surface by the way of Fc receptor. Human express 3 different Fc gamma and 2 different Fc epsilon receptors, each of which has a characteristic tissue distribution and biological properties. The high affinity receptors of each type, Fc gamma receptor 1, Fc epsilon receptor 1, bind immunoglobulin under normal conditions and so produce the notable biological effect. The physiological function of Fc receptors differ among cell types. Binding of secreted immunoglobulin G molecule into Fc gamma receptors on macrophages or natural killer cell serve arm to these cells to carry out ADCC reaction. Fc receptor facilitate phagocytosis of antibody-coated particles through the phenomena called opsonization. They are important for triggering chemotaxis and degranulation in neutrophil and other phagocytes. Binding of antigens to Fc epsilon receptor 1 associated immunoglobulin E on cells like basophil and mast cell provides the signal for degranulation and can lead to allergic symptoms. In contrast, low affinity Fc receptors in B lymphoid cells enable these cells to sense local antibody concentration, so provide an important feedback pathway that limit further antibody production. Biologic activities of immunoglobulins. Immunoglobulins are bifunctional molecules that first bind to antigens. This is characteristic by specificity and diversity. Second, can initiate other biological activities that are critical for host defense like opsonization, complement activation, and crossing placental barrier. The first functional is localized as a combination of variable heavy and variable light domain, while the second function is related to the constant heavy domains, particularly those of the Fc segments. First type now we are going to talk about different types of immunoglobulins. Account for 75% of total serum. It accounts for about 75% of total serum immunoglobulin. It's produced during secondary humoral immune responses. It is the only class of immunoglobulin that crossed the placenta. Immunoglobulin G2 is less efficiently transferred than others. Antigen-bound immunoglobulin G is capable of fixing serum complement with different degrees. C1q binding site on the immunoglobulin G proteins found in constant heavy chain 2 domain. Immunoglobulin G4, it is unable to fix complement via classic pathway, which require binding of protein C1q. However, it may be active in alternative pathway. Macrophages and certain other cells express surface receptors that bind the Fc regions of immunoglobulin G molecules. These receptors interact with constant heavy 3 domain. They bind immunoglobulin G1 and G3 with much higher affinity than they bind to other subclasses. The second type of immunoglobulins is immunoglobulin A. It is the predominant immunoglobulin produced by B cells in various batches, tonsils, and other some mucosal lymphoid tissues. It represents only about 10 to 15% of serum immunoglobulins. It is abundant in saliva, tears, intestinal mucus, bronchial secretion, milk, and prostatic fluid, and other secretions. On B cell surface, immunoglobulin A exists as a monomer composed of 4 chain units, but in the blood or secretion, it forms polymer of up to 5 such units that are associated with 1 molecule of the chain and secretory component. The two classes, immunoglobulin A1 and A2, are expressed at ratio 5:1 in blood and have similar properties. This is the structure of immunoglobulin A. Immunoglobulin M. It represents about 10% of normal serum immunoglobulins. It predominates in early primary immune response to the most antigens, but tend to be less abundant subsequently. It is the class most frequently found as natural antibodies against certain antigens, blood grouping determinant, usually combined by immunoglobulin D on the surface of B cell, particularly the virgin B cell. It is the most efficient complement-fixing immunoglobulin. A single molecule of antigen bound to the immunoglobulin M is sufficient to initiate a complement cascade. This is the structure of immunoglobulin M. It is secreted as a J chain containing pentamer. This is the structure of immunoglobulin M as a pentamer. Immunoglobulin D. It is a monomer with molecular weight about 180,000, commonly found on the surface of B cell, and only traces are found in the serum. In cells that express immunoglobulin D and M, immunoglobulin D can bind antigen and transmit signals to the cell interior, with consequence that appears to be identical to those produced by immunoglobulin M. When such B cells become activated, surface immunoglobulin D expression decreases. It is relatively degraded by heat and proteolytic enzymes. It has antibody activity toward insulin, penicillin, milk protein, diphtheria toxin, nuclear component, or thyroid antigens. This is the structure of immunoglobulin D. Immunoglobulin E. It represents only a very small fraction of serum antibodies. Immunoglobulin E exists only in a monomeric form. Immunoglobulin E is involved in allergic disorders. Elevation the levels of serum immunoglobulin E may be also a signifier for infection by helminthes or other types of multicellular parasites. This picture explains the role of immunoglobulin E in histamine secretion by mast cell. Mast cell or basophil bind immunoglobulin molecule after they are secreted by activated B cells. The soluble immunoglobulin E antibodies bind Fc receptor proteins on the mast cell surface that specifically recognize the Fc region that found in immunoglobulin E antibodies. The passively acquired immunoglobulin E molecule on the mast cell surface serve as a cell surface receptor for antigen. Thus, unlike B cells, each mast cell has a set of cell surface antibodies with a wide variety of antigen binding sites. When an antigen molecule binds to these membrane-bound immunoglobulin E antibodies so as to cross-link them on their neighbors, it activates the mast cell to release histamine by exocytosis. This is the structure of immunoglobulin E. Mechanism of antigen elimination by antibodies. First, toxin neutralization or virus neutralization, opsonization, phagocyte activation, activation of complement, antibody-dependent cell-mediated cytotoxicity. Thank you.